challenge. Hits go like ooh, ooh, hey, hey, turn up. Ooh, ooh.
Right, first, let's get started in DNA replication. As you all know, DNA is vitally important for every living organism. It controls the movement and the action of the cell. It also decides the physical and the genetic trait of that organism. Basically, the DNA is what makes us us. It is important for the cell and required to be duplicated for the other cell during replication. And that process is called DNA replication. It always happens during the interface of the cell cycle for obvious reasons. So, let's talk about the replication process. Here is the original DNA. Before anything can happen, the helical will unzip the DNA into two separating strands. One is called leading strand, the other one is lagging strand. I will explain further later. Leading strand. So what do we have here is called a primase. Its job is to lead the DNA polymerase by adding starting signal, meaning that the DNA polymerase have to start from the signal till the end. Adding complementary bases, adenine with thymine, guanine with cyanide. Lagging strand. It is a bit different and more complicated from the leading strand. I explain how. Remember that polymerase only can work from 5' to 3' direction, meaning that it is impossible to work from 3' to 5'. However, it is still able to move in that direction without working. Right, so let's talk about the process as a whole. So, this is the lagging strand, and I have indicated 3' and 5' for easier to understand. The DNA primer will mark the starting codon throughout the DNA in multiple positions. Because the polymerase only can work from 5' to 3', and meanwhile it also has to move from 3' to 5' to follow the helicase. Here's an animation for you to easier to understand. First, the primer is working in codon number 1 from 5' to 3'. Now, it moves in the opposite direction because it has to follow the helicase. It will again start working in the second codon, and the process will repeat itself until completion. These are called the Okazami fragments. This is the bigger picture for you. When completed, there will be two DNAs ready for service. Each DNA is mixed with one strand from the original, and the other one is from the replication. It is now ready to use during the mitosis. Now, Eric will talk about the protein.